Hello everyone, my name is Diane Webster. I'm the Research Director for the Prader-Willi Research Foundation Australia. It's an absolute pleasure to join you all today, and I'd like to thank the organisers for the opportunity to provide you with an update on current PWS research. Before we begin, I'd like to acknowledge the Wurundjeri people of the Kulin Nation as the traditional owners of the land from which I'm joining you, and pay my respects to all elders throughout Australia, past, present, and emerging. At the Prader-Willi Research Foundation Australia, our goal is to find, grow and support high-quality research that focuses on therapies for Prader-Willi. Whether it's improving existing therapies and their delivery, research that targets symptoms, or long-term studies that target the underlying epigenetic cause of PWS, our objective is to bring about meaningful change for people with PWS and their families. The Foundation's strategic research framework ensures that the research we fund is balanced and therapy-focused. Our talented and multidisciplinary Scientific Advisory Council, chaired by Professor Joe Pareto, ensures that every Foundation-funded project meets the highest scientific standards. We aim to support the most powerful science that Australia has to offer to address the most pressing challenges that people with PWS face. Right now is a really exciting time for PWS research. We're so close to not one but two therapies for hyperphagia, with levotherapeutics and selenotherapeutics working through the drug approval process with the FDA in the US. The number of clinical trials for PWS is also expanding and covering an increasing list of symptoms, including hyperphagia, anxiety, exercise, sleep, temper tantrums, and bone sensitivity, to name a few. Research is accelerating. There are more scientists than ever before working on the unmet needs of the Prader-Willi community. In preparation for this talk, I looked over the Prader-Willi papers that have been published in the last 12 months. What's really striking is the diversity of topics that have caught the imagination of researchers. For this talk, I've grouped them into three themes. The first theme is about capturing the Prader-Willi lived experience. Much of this research is driven by the Global PWS Registry, and it shows the power of working at a global level to amplify our voices. We've seen key reports that document the patient voice, put numbers around caregiver burden, and a study from Australia that looks at the costs associated with PWS. There are also reports that clearly articulate the PWS community's thoughts on treatment priorities, and really importantly, on the amount of risk or side effects that would be considered acceptable in exchange for an effective therapy. What's so powerful about this research theme is that it's embedding the voices of people with PWS into a robust, peer-reviewed scientific context. Essentially, it's translating your lived experience into scientific currency. That's important because it allows people with PWS and their parents and carers to help set research priorities. Scientists use these papers to convince funding bodies that what we're doing is important. Regulators and policymakers use these papers to better understand how important therapies are and what impact they will have. Finally, in this theme, I'd like to highlight a paper by the PWS Clinical Trials Consortium. They reported consensus definitions for common PWS symptoms like hyperphagia, temper outbursts, and anxiety. This work helps ensure consistent and clear communication, and it's fundamental to effective clinical trials and the delivery of healthcare. The second research theme I've called Behind the Scenes. These studies have been unpacking the basic science behind PWS. We all have 2 copies of chromosome 15, one from mum, one from dad. The Prader-Willi region of mum's chromosome 15 is imprinted. That means these genes have been silenced or turned off, while the genes on dad's chromosome, they remain active. In people with PWS, dad's active genes aren't working. They've either been deleted, or you've ended up with 2 imprinted copies from mum, that's UPD, or perhaps something went wrong in the imprinting process. The result is that all of the proteins and RNAs that you can see coloured here on the slide in blue are not being made when they should be. Over the last decade, we've learned a huge amount about how this part of the chromosome is regulated. Specifically, what's keeping mum's genes silenced. A number of scientists are now working to find ways to turn these important genes on as a potential therapy for PWS. For example, in the last year, scientists in the Chamberlain-Leland lab in the US have used CRISPR-Cas9 to delete critical binding sites in the SNORD116 locus. They were able to activate expression of SNORD116 RNAs, and that's confirmed that targeting these binding sites is a potential therapeutic strategy for PWS. My favourite paper from the last 12 months is from Professor Marnie Blewett's lab. It also reports an important advance in activating PWS genes. The Prader-Willi Research Foundation Australia have been working with Prof Blewett and her team for a number of years now. Their research is helping us understand how an enzyme called SMooCD1 is keeping the MAGEL2 cluster of Prader-Willi genes silent. Specifically, this paper unpacks the role that SMooCD1 plays in establishing and maintaining imprinting of the MAGEL2 gene cluster. What's really exciting about this research is that it suggests that SMooCD1 does most of its job early in development, establishing imprinting of a number of different gene clusters while babies are in utero. But after that, it's only needed by a small number of genes to maintain imprinting, including the genes at the MAGEL2 end of the Prader-Willi cluster. This is an important finding because it suggests that after birth, it should be relatively safe to target SMooCD1 as a way of activating PWS genes. On this slide, you can see that SMooCD1 is keeping the MAGEL2 gene cluster switched off. The Blewett lab hypothesis is that removing SMooCD1 will turn the MAGEL2 cluster back on. So far, they have shown that removing SMooCD1 in mice, by genetically knocking it out, does activate gene expression, and that's great news. What they're working on now is finding a small molecule inhibitor that will do the same thing, but in the same way the drug works. To find that potential new therapy, the Blewett lab have recently undertaken a high-throughput screen of 290,000 small molecules or compounds. From this screen, they have identified over 2,500 potential SMooCD1 inhibitors, or hits. The next step, which is being done right now, is to validate those hits. So they take each hit from the high-throughput screen and they repeat the same assay, but at lab scale. What they're looking to do is check that that interaction was real and not just an artifact of the large-scale screening. Any positives from that screen are then put through a separate or a different assay to see whether they can have the same effect. So in this way, they validate that their results are strong and positive. The information from all this testing enables the scientists to select lead compounds. These are the small molecules with the most promising profile, those that show the best specificity and selectivity for inhibiting SMooCD1. It's important to note that although these top 5 or 6 compounds can't yet be considered drugs, they are the precursors to drugs. They will enter a medicinal chemistry program to refine and optimize their structure and function, and that's pretty exciting. Now let's dig a bit deeper behind the scenes and consider some of the advances that have been made in understanding the form and the function of the genes in the PWS region and the downstream pathways which may be dysregulated in PWS. The graphic I've chosen here is to help demonstrate that each gene in the PWS region has a job to do, and it creates a very complex picture. My take-home message is that every year we learn more about these genes and their pathways, and that's really important for 2 reasons. Firstly, lots of these pathways have already been targeted for drug development, raising the possibility that we can repurpose a medication from another condition to PWS. And secondly, it supports rational drug design, which is where researchers can design a drug for a specific target. Prof Blewett's SMooCD1 inhibitors are an excellent example of this process. So looking at some of the recent advances in basic science over the last year, I'd like to highlight a study from the Webster lab in Canada, where they've been investigating the role of the N-terminal, or the front half, of the MAGEL2 protein. They report that this region of the MAGEL2 protein is an intrinsically disordered protein, which has a potential role in RNA metabolism. What's really fascinating is that disruption of intrinsically disordered proteins has also been associated with autism spectrum disorder. So this has the potential to add to our understanding of a specific PWS phenotype. In other research, the SHAFE lab have undertaken molecular studies which have shown that the mTOR pathway is upregulated in SHAFE-Yang syndrome. The mTOR pathway is involved in metabolism and is also dysregulated in diabetes and obesity. As a result, the SHAFE lab are now investigating rapamycin in cell culture and mouse models as a potential therapy for both SHAFE-Yang syndrome and Prader-Willi syndrome. There have also been several studies looking at topics like brain architecture, neuron connections, hypothalamic dysfunction, and various other pathways and gene interactions, many of which are or may lead to druggable targets with the potential to ameliorate symptoms of PWS. The final research theme is clinical studies. These are studies that seek to better understand and characterize the signs and symptoms of PWS and may also evaluate potential interventions. It includes major clinical studies such as LV101 or carbetocin from Levo Therapeutics and DCCR from Soleno Therapeutics. Both of these potential therapies have been able to achieve and maintain a substantial decrease in hyperphagia for many trial participants, as well as improvements in various behaviours and anxiety measures. LV101 is currently being reviewed by the FDA, with an outcome expected in the near future. The pathway forward for DCCR has been more complex. However, they have recently reported promising data which compares their open-label extension study to a matched cohort from the Path for PWS study, which is a promising development. If you'd like to know more about either of these trials, you can find recordings of each company's presentation from the recent FPWR conference on their YouTube channel. In the last 12 months, we've also seen the publication of results from 4 microbiome randomized placebo-controlled clinical trials. All 4 studies showed that the trial probiotic was safe, but only one was able to demonstrate any impact on the PWS phenotype. They found a very small decrease in BMI, along with improvements in social communication and fine motor skills. This is a promising start, but the changes being reported are more about polishing and fine-tuning than the sort of wholesale change that we need to see to really have an impact on life with Prader-Willi syndrome. There have also been a large number of growth hormone studies published, but most have reported confirmation or small extensions on previous work. It's also worth highlighting that there's an increasing number of clinical trials currently recruiting or planning to start next year. Beyond clinical trials, there were also some really interesting clinical observation papers published in the last year. This type of work is really important because it can lead to biomarkers that we can measure, monitor, and in some cases alter. For example, Damien et al. Investigated cortisol in hair and found that when combined with other measures, hair cortisol concentration might be helpful in diagnosing adrenal insufficiency. In another study, Frienza reported a possible link between vitamin D and the hormone irisin, which has been associated with bone mineral density and cognition. Now, while both of these studies do still require additional work, they offer insights into the diverse phenotype of PWS, and they may lead to personalised treatments and better health outcomes in the future, as do a number of other studies which have looked at sleep, exercise, hormone variants, and many other aspects of PWS. Importantly, these types of studies can also uncover opportunities. They can point to new pathways and targets that can be used to develop new therapies or repurpose existing therapies. To bring all this together, let's take a look at the typical drug development pathway. In this representation, we move from a basic or a clinical science observation through to a target identification, validation, and optimization. These are the steps by which an observation or a lab result is turned into a potential therapy. Potential therapies can enter this pipeline at almost any stage, and they may even skip steps. So if we look, we've got an example here for rational drug design. That would be work such as that from the Blewett lab. But you might also have an observation that lends itself to a non-drug intervention. So an example here would be mindfulness studies, such as the trials being run by Lauren Rice and Stuart Einfeld. So something like this might start with a clinical or apparent observation and then move quite quickly to a clinical trial. It's also possible to join this pipeline at the late preclinical or the clinical trial phase. You'll often see companies doing this, where they have a molecule, a drug, such as Neuron Pharmaceuticals NNZ-2591, where they've tested this already and optimized it. They've got a drug formulation. For example, with Neuron, they work in other neurodevelopmental disorders, and they're looking around for other conditions that might also benefit. This has, as I've said, happened with Neuron earlier this year, where they added PWS to their pipeline. They did a small preclinical trial in a mouse model, got promising results, and they're now planning for a phase 2 trial next year. So you don't have to start right at the beginning. You can actually join the pipeline further through. The final arrow here is to represent repurposing. This is where a drug has gone all the way through this pathway, is out the other end and in the market, and we've identified some function of that drug which we think might be useful in Prader-Willi syndrome. So it's currently licensed for another indication, and we're going to repurpose it, probably funneling it back in with some case studies or a small clinical trial to show efficacy in Prader-Willi syndrome and, of course, safety as well. This pipeline is also a good way to consider how the Prader-Willi community might engage with research. PWS research can't and indeed shouldn't happen without you. This goes far beyond fundraising for research. So while that is important, it's only one of the many ways you can be involved. What's vitally important is that the voice of people with PWS is represented at every stage of this research process. By sharing your stories, you can help inform research priorities. You can do this by talking to your clinician about what's important to you, or by joining a registry or a biobank. Strong and genuine partnerships with the PWS community can encourage small companies like Neuron to add PWS to their product development pipeline. In addition, project-specific focus groups, steering committees, and advisory bodies all need patient advocates to inform and improve trial designs. You can also have a look around and find out which clinical trials are enrolling in your area and considering enrolling in a trial if it looks like a good fit for you. Finally, by sharing your stories, we can paint a real-life picture for regulators and policymakers and influence decisions that can have a real impact on the lives of people with PWS. To finish today, I'd like to introduce our Chromosome 15 Biobank. The Chromosome 15 Biobank is a joint initiative established by Associate Professor Godler, Professor Amer, and Dr. Baker at the Murdoch Children's Research Institute in Melbourne. It was established earlier this year in partnership with PWRFA and FAST, and we'd like to acknowledge funding from the Lawrence G. and Jean E. Charitable Trust. The biobank is currently recruiting. They are collecting and storing blood and buccal samples from people with chromosome 15 disorders. The biological samples will be linked to clinical histories and deep phenotypic data. This information is being used by Associate Professor Godler's lab to better understand the link between phenotype and genotype, specifically in the area of mental health. This exciting research is already pointing to some new pathways that could be targeted in the development of new therapies. But the real power in a data-rich biobank such as this is its accessibility to other researchers from both academic and industry settings. If you're interested in finding out more, please contact Emma Baker, whose address is on the slide. And with that, I'm happy to take questions.